Hello friends, welcome to Sam Biology class. This is Subrat Kumar Mahanti. Today we shall discuss about gene mapping by somatic cell hybridization. In addition to deletion mapping, genes can also be mapped by somatic cell hybridization. Somatic cell hybridization, which involves the fusion of different types of somatic cells. Most of the mature somatic cell can divide only for a limited number of time, because of which such type of cells cannot be grown continuously in laboratories. But certain cells, such as those have been altered by virus or those are derived from tumor, can be grown continuously because the cells have lost control over cell division, because of which such type of altered cell divide continuously in order to produce a cell line. In somatic cell hybridization, cells from two different cell lines are fused in the presence of polyethylene glycol or any other agent which alter the plasma membrane. So here these are cells of two different cell lines. Two different types of cells are fused in the presence of polyethylene glycol or any other agent that alter plasma membrane. This alteration in the properties of the plasma membrane makes it possible for two different types of cells to fuse together. So this fusion results in the formation of a cell which has two genetically different nuclei. Such a type of cell which has two or more than two genetically different nuclei is known as heterokaryon. Heterokaryon. And finally, these two nuclei also fuse together, that results in the formation of a somatic hybrid cell. That results in the formation of a somatic hybrid cell. That results in the formation of a somatic hybrid cell. For example, human and mouse somatic cells can be fused in the presence of polyethylene glycol to form a human-mouse somatic hybrid cell. This hybrid cell will possess the chromosomes of both human and mouse. But as these hybrid cells divide, they tend to lose some of the chromosomes, preferentially from only one species. In case of human-mouse somatic hybrid cell, some of the human chromosomes are preferentially lost and remaining are retained in the hybrid cell. After the loss of chromosomes, eventually the hybrid cell is stabilized. This loss of chromosome from the hybrid cell is random and differ among cell lines. This property of retaining extra human chromosome in the hybrid cell makes it possible to determine the location of specific genes on a chromosome. Gene mapping by somatic cell hybridization requires a panel of different hybrid cell lines. This figure shows a panel of 6 cell lines, each of which is produced due to the continuous division of a human-mouse somatic hybrid cell. These hybrid cells contain all mouse chromosomes plus few human chromosomes. The cell lines are then examined microscopically and the human chromosomes that they have retained is identified. The cell lines of the panel have been chosen so that each of them will contain a different subset of human chromosomes. For example, cell line A possesses human chromosomes 2, 4, 7 and 8. Cell line B contains another different subset of human chromosomes that include chromosome 1, 2, 4, 8, 9, 10, 11, 12 and 13. Other hybrid cell lines also possess unique subsets of human chromosomes. The cell lines are then examined for the presence of a particular gene. The gene can be detected by looking for the gene itself or for its product, for example, an enzyme. In this example, we want to determine the location of a gene that encodes a particular protein, for example, an enzyme. Out of 6 cell lines A, B, D and F possess the gene product. And comparing the different subsets of human chromosomes, it becomes clear that cell line A possesses human chromosomes 2, 4, 7 and 8, whereas cell line F contains human chromosomes 4, 19 and 20. Both of them possess the gene product. The chromosome which is common to cell line A and F is chromosome 4. That means the particular gene is located on human chromosome 4. In this way, the location of specific human genes can be determined on various human chromosomes. So the correlation between the presence of a specific gene and the presence of a specific human chromosome makes it possible to determine the location of a number of genes on the genome. In addition to this, somatic cell hybridization can also be used to position a gene on a specific part of a chromosome. Some hybrid cell lines carry a human chromosome with chromosomal mutations such as deletion and translocation. When a gene is present in a cell line with intact chromosomes but is absent from another cell line with chromosomal deletion, then the gene must be located on the deleted region of the chromosome. Let us compare between cell line 1 and cell line 3. In cell line 1, all chromosomes are intact, but in cell line 3, chromosome 4 has a deletion. Because of this deletion, the gene has been removed from this chromosome. So this proves that the gene is located on the short arm of the chromosome 4. Similarly, when a gene is absent from a chromosome but consistently appears whenever there is translocation, then the gene must be present on the translocated region of the chromosome. So friends, this is all about gene mapping by somatic cell hybridization. For more updates, subscribe my YouTube channel, Sam Biology Class.